Hello students. So in this video, we look into the selection and screening of recombinants. So we have known the steps involved in RDNA technology. So it involves a conventional restriction and ligation cloning protocol. So the major four steps being fragmentation of the DNA with the restriction endonucleases, ligation of the DNA fragments to the chosen vector. So in this case, a plasmid vector, or it might be a phage vector or a hybrid vector, or it might be an eukaryotic vector. Then introduction into the bacterial cells of the recombinant vector into the bacterial cells by transformation, and then selection and screening of recombinants. So the success of a cloning experiment here depends on the ability to identify the desired gene sequence among the many different recombinants that may be produced. So you have a host of recombinants, and from among them you will have to screen your recombinants. So identifying a clone can involve using some sort of selective mechanism, right? So here we call it as a selection. That is, you're providing a selective pressure in the medium itself, so that this allows the or it confers the ability only the recombinants to grow and the non-recombinants to be inhibited. Or it can be a screening methodology where you screen a population of clones with a specific probe. So it can be a radioactive cDNA or a oligonucleotide. So these two are the major methods of identification of your recombinants. So it can be selection or screening. So selection gives you a ability of the recombinants to be growing, or it gives an advantage over the non-recombinants. Whereas screening, from a library of clones, you are selecting or you are going to identify only the recombinants from them through some sort of probe. So to continue with, in this video, we'll be dealing only with the direct methods, right? So that is, I mean, the selection of recombinants. So in this video, we'll be only discussing about the direct methods of your selection of recombinants. The first method being visual screening. So here, this can be the blue and white screen is the most popular system, yet one another method is the use of antibiotic resistant marker. So we know that the plasmids have a antibiotic resistance as a marker. Fine. So if you're going to clone your foreign DNA segment in a plasmid vector which carries an antibiotic resistant marker, so what happens is the recombinants alone will be now be resistant to a particular antibiotic, the marker which you have used, while the non-recombinants will be sensitive to the antibiotics. So this is one methodology. So visually you are differentiating between the recombinant and non-recombinant by means of the antibiotic. But the most popular system here which we are going to discuss is a blue-white screening. So this method utilizes the chromogenic substrate, right? So here the chromogenic substrate is X-gal, which is 5-bromo, 4-chloro, 3-indoloyl, beta-D-galactoside. So this substrate is now broken down in by the beta galactosidase. So we are now relying on the ability of a activity of the functional beta galactosidase enzyme. So this enzyme beta galactosidase is a product of a lag acid gene of the lacopron. Now, it is a tetrameric protein, and you can have the subunits like an initial N-terminal region of the protein. That is what we are going to be like. It's a one the key for the activity of the protein. So, what do you mean by this here? You are going to use a host which is a mutant. So, the host here which you are going to use contains, right, only a part of the laxi gene. So it is a gene, lac is a gene without the initial region. Fine. Whereas the vector, that is the plasmid which you are going to have, is going to contain the alpha peptide to complement the defect to form the active enzyme. So what does it mean when you have a mutant host which is lacking the initial region of beta galactosidase, and the vector which is going to have only the alpha peptide region of the enzyme. So what happens when you are going to put them together, when there is, after the transformation, when there is a complementation. Okay, so that these genes complement, we call it as gene complementation. There is complementation so that there is a functional active enzyme, that is beta galactosidase. As a result, what happens if a vector containing the alpha peptide will be transformed into the host containing the remaining lac acid gene, the two fragment will reconstitute to form the active enzyme. In addition, the alpha peptide region in vector contains multiple cloning sites. So we'll be talking about the following the insertional inactivation. So this can be utilized in insertional inactivation also. But now for time being, let us discuss the function of a complete beta galactosidase enzyme, right? So the host is mutant and it carries only the carboxypeptide region of the enzyme, and the vector contains the genes only for the alpha peptide region of the enzyme. So when these are put together, there is gene complementation, and what happens? There is a functional beta galactosidase being synthesized only in a recombinant. Okay, that is the cells which are carrying the plasmid vector, fine, which provides the alpha peptide region of the enzyme. So this functional beta galactosidase enzyme will oxidize this X-gal in the presence of the IPTG. Isopropyl thiogalactosidase is a inducer. So in the presence of IPTG, the beta galactosidase will oxidize X-gal to form 5-bromo 4-chloro, right, endoxyl galactose and galactose. I'm sorry, 5-bromo 4-chloroendoxyl and galactose. Now, this endoxyl derivative is oxidized in air to give a blue colored dibromo dichloro derivative. So that gives you the blue color. Okay. So a colorless substrate is now, upon action by the colorless substrate, here we mean X-gal, upon action by beta galactosidase is converted into a colored product, which is blue colored, right? The dibromo dichloro derivative, which is blue colored. So the blue colored colonies indicate the absence of insert, whereas colorless colonies indicates the presence of an insert. So in case you are utilizing this for insertional inactivation, right? So if you're using this for insertion inactivation, for example, right? So we'll look into the here the illustration, then we'll have a better understanding of this. So you have, we start with the vector PUC18. So it carries an ampicillin resistance and NCS. The lac is a gene, okay, and there you have the MCS. So you're cloning your foreign DNA in this MCS region. So what happens? There are two options. There are two possibilities. One is without the insert, the other with the insert of the foreign DNA. So as you can see now in this diagram where there is no insert, you can see that now what happens, there is no insert, and now as you can see there is no insert. So the lac is a gene. Here you can see a functional alpha peptide region is synthesized. Sorry, the carboxy portion. I'm sorry, the carboxy portion is synthesized by the host. It lacks the alpha peptide region. Now the vector is going to synthesize the alpha peptide portion of the enzyme, the beta galactosidase. So there is gene complementation. Now we have the functional beta galactosidase enzyme. Since there is no insert into this region of the foreign DNA. Here in this case, now what happens? You have the foreign DNA being inserted. So this is disrupted. The gene is disrupted. So here there is no production of the alpha peptide region of the enzyme. Only the carboxy portion of the enzyme is there, so which is now a non-functional or incomplete enzyme. So it doesn't make up a functional beta galactosidase. Now here, so without the insert, you have a functional beta galactosidase. With the insert, you have a non-functional enzyme. Now these are the two possibilities which can occur during your experiment, and after transformation, how do you screen them? So you're going to plate both of them together. You're going to plate all the cells on a X-gal. So that is your 5-bromo 4-chloro 3-indoloyl beta D galactoside. So what happens? the cells which are synthesizing the functional beta galactosidase, fine, they will cleave the X-gal to give a blue colored product. Whereas the cells without the insert, right, that is a recombinants. What do you mean to say? The non-functional enzyme, the cells which do not make a functional beta galactosidase. Why they do not make a functional beta galactosidase? Because they're having the foreign DNA disruptor, okay, into the lactazid gene. So they do not make a functional beta galactosidase. So those cells will be white in color. They do not give a blue color. So now you can differentiate them on the medium itself. As you can see, I've given you the picture of a plate, microbial culture plate, which shows you the differentiation between the blue colored and the white colored colonies. So the blue colored colonies are non-recombinants, whereas the white colored colonies are recombinants. Now we move on to another topic, right? Another phenomenon, insertional inactivation. So it is defined as a marker, whereas insertional inactivation, it's a phenomenon where the marker gene function of a cloning vector is inactivated due to the insertion of foreign DNA. So we'll see the insertional inactivation of various marker genes. So in this case, antibiotic resistance, right? So if an antibiotic resistant gene is disrupted, fine, by a foreign gene, what happens? The cells which carry the do not carry the cells which do not carry the foreign DNA insert, okay, the carrying the cloning vector, which is not having a disrupted marker gene, they will be resistant to a particular antibiotic, whereas the other, that is the recombinants, will be sensitive. So in that case, you need two antibiotic marker genes. Let's say the simplest one first, in the case of antibiotic resistance. Now, as you can see, with the case of a single antibiotic resistant marker, okay, the cells are grown and you can replicate onto the medium, one which contains ampicillin, the other which doesn't contain the ampicillin. So here you mean to say that the cells which are growing, right, on an ampicillin containing medium are the recombinants. Now, this is a case when just a single antibiotic marker is utilized. So, this is a visual screening example. Now, if in the case of insertion inactivation, you need at least two antibiotic markers. Now, for PBR 322, that is a classical example. You have two resistant markers, one tetracycline, the other is a ampicillin. So, if you see, one of the antibiotic resistant marker can be utilized. Say for example, if you have now cloned in the place of a tetracycline resistant gene, right? You grow these recombinants, the transformants on a medium containing ampicillin. So both recombinants and non-recombinants grow. Later, if you do a master plate, similar to this shown on the illustration, if you do a master plate technique and transfer to a tetracycline containing plate, only those cells which are carrying this tetracycline resistant gene, okay, so that is the tetracycline sensitive cells will not be growing, whereas tetracycline resistant cells will be growing. You compare both the plates and you select only the cells which are resistant to ampicillin and sensitive to tetracycline. So this is a case with the insertional inactivation with the antibiotic resistant gene. Now if you are using a phage vector, okay, so if you are using a phage vector, the gene that is chosen is your C1 gene. Now this C1 gene is a one which decides whether a lytic or lysogenic stage is undergone by the virus in a bacterial host. So the C1 gene encodes for C1 repressor and it is responsible for the formation of lysogens. In the presence of a functional C1, the plagues, that is the growth of a plague or the growth of the virus on a bacterial cell. Fine. So the plagues contains unlysed host cells and has a turbid appearance, whereas in the absence it will be clear. So here, when there is insertional inactivation of the C1 gene, right? So what happens? There is no repressor being synthesized and all the cells will undergo a lytic stage or the phase. Fine. So Here you can differentiate by means of the turbid and the clear plaques. So here you have the illustration which shows you the differentiation. Okay, so if you're insertionally inactivating the C1 gene, what happens? You get a clear plaque. So the turbid plaques or the non-recombinants and the clear plaques are the recombinants. Fine. Now, another methodology of direct selective selection pressure is your complementation of mutation, which you can use with that of your yeast vectors when you're utilizing yeast vectors. So we know the yeast vector genes carry certain genes like histidine 3, leucine 2, TRP1, and URA3. These are the selectable markers which are utilized. So in this approach, again, you need a mutant host strain. Okay. So the mutant host strain now contains a missing gene. Fine. So it might be the missing histidine gene, missing leucine, missing tryptophan, or missing uracil 3 gene. And the gene is now complemented or provided only by the vector. So the transformants will grow only if the gene product from this clone will complement the function. So the medium that you'll be utilizing is a minimal medium. Fine. So what happens? The non-recombinants cannot synthesize them on their own, whereas recombinants which carry the functional selectable markers, they will be able providing or to confer the ability to the growth. Okay. So the yeast vector host with a mutation in these genes are available and can be used with the yeast vector to screen the recombinant clone. Okay. So here, in this case, the uracil 3 and the leucine 2 marker offer both positive and negative selection. Now, what do you mean by a positive selection? In the positive selection, the host strain doesn't grow on the media, lacking the functional gene. Okay, that is on a minimal medium. They will not be having the ability to grow. They will not be having the ability to synthesize the missing component, and they will not grow on a minimal medium. But the host transformed with the recombinant clone, that is these marker genes, any of this, the uracil and the lysine, uracil and leucine 2, uracil and leucine 2 markers, okay, with the recombinant clone, they can be able to supply the gene product and required to grow in this minimal media. So this is a positive selection. It gives you a selective pressure where only the recombinants grow. The non-recombinants do not grow. Now we'll move on to the next, the negative selection. So in negative selection, a chemical compound is added to the media which will be converted to the cytotoxic agent in the presence of the gene product. And as a result, it doesn't allow the growth of the wild type cells. Okay, so let us take this example. Fine. But of uracil 3. But here what happens, a host strain which is transformed with the recombinant clone has a non-functional gene product and grows in the presence of the compound in the media. Fine. So here let us say the negative selection of uracil 3 marker gene. So ERA3 is going to code for orotyne 5 monophosphate OMP decarboxylase. And this active enzyme will process the 5-fluorouric acid to the toxic fluorodeoxyuridine. Okay. So the component present in the medium will be the 5-fluorouric acid and it is converted into the toxic fluorodeoxyuridine. So if you are going to have this as a marker gene. So generation of this cytotoxic agent kills the cells carrying the functional uracil 3 gene product. Okay. So if you are insertionally inactivating it, okay, if you are having going to have a foreign gene insert, what happens? Obviously, this enzyme is not synthesized and it is going to have the ability to grow on this medium. Whereas if it is carrying a functional uracil 3 gene, what does it mean? It doesn't carry the foreign gene insert in that. So it is now killed. So only the recombinants survive on the medium. So this is one mode of a negative selection. Now, last topic of this lecture is your reporter gene assay. So here we are going to talk about the vectors and the vectors which are being used, the assay methods which are being used for eukaryotic mammalian cell lines. So here we are talking about reporter gene assay. So the mere presence of a gene, a reporter gene, will indicate the presence, it is going to report or indicate the presence of a recombinant. So here we have a chimeric construct, which is carrying a promoter, eukaryotic promoter, and the reporter gene, which will code for an enzyme, which converts the substrate into a product, which is easy for us to read out. We are going to talk about two types of reporter gene assay in this video. The first one, luciferase reporter gene system. So luciferase is an enzyme present in the abdomen of the firefly tonus of pyralis. Okay. So the enzyme utilizes the D-luciferin as a substrate to form oxyluciferin. In the presence of ATP Mg2+, luciferin is again converted into the luciferin adenylate, involving pyrophosphate cleavage and transfer of AMP to luciferin. So this luciferin adenylate undergoes oxidative decarboxylation to oxyluciferin with simultaneous emission of light. So I repeat here, this substrate luciferin is converted to oxyluciferin. How? In the presence of ATP Mg2+. So first, luciferin is converted into luciferin adenylate, which is involving the pyrophosphate cleavage. That is, ATP is cleaved, and then the AMP is transferred to luciferin. Now this luciferin adenylate will undergo oxidative decarboxylation to oxyluciferin. So there is emission of light. So this is schematically illustrated. The reaction is illustrated over here. Fine. So luciferin in the presence of ATP and Mg2+, you have the oxyluciferin, which gives you a light emission. So this can be screened or measured. So this is a first reporter gene assay we have talked about. The second one which we'll be talking about is the most popular one, GFP, green fluorescent protein. So in the live mammalian cell, the green fluorescent protein will be a better choice as a reporter gene to screen cells containing the recombinant protein. So they are fluorescently tagged, okay, at the C terminus. The cells receiving the recombinant DNA will give the green fluorescence, and it can be visualized with an inverted fluorescence microscope. Alternatively, you can also go for flow cytometric analysis. The flow cytometer analysis, the cells based on its size, shape, and fluorescence level, all these. Okay, so flow cytometer will analyze the cell based on its shape, size, and fluorescence. A non-fluorescent cell gives a separate peak as compared to the fluorescently labeled cells, and we can compare these on a graph. So here you can see the green fluorescence, right, under the inverted fluorescence microscope, and also the flow cytometric analysis. So here you can see two different, like you can see the two different peaks. This is a peak where you don't have the fluorescence, that is non-recombinant. So here the green fluorescence given out by the recombinants. Fine. So there are various other reporter genes apart from green fluorescent protein. You have a red fluorescent protein, you have a yellow fluorescent protein. You can also use a beta galactosidase, beta lactamase, the chloramphenicol acetyltransferase, luciferase. These are the other additional reporter genes which are being utilized in your eukaryotic cell lines. Okay, and the various applications are listed as you can see: in vitro drug screening, intracellular drug screening, high throughput screening, in vivo parasite monitoring, the whole animal or organ imaging in the case of the green fluorescent or the red fluorescent proteins, the in vivo drug screening, vaccine efficacy testing, gene expression studies, protein co-localization, how a protein is localized in a particular cell, you can study them. So these are the various applications of the reporter genes which are being listed, right? So I conclude with this, and to sum up, we have seen that the most successful, right? So the most successful screening, okay, the most successful step of a recombinant DNA technology is the selection and screening of the recombinants. So selection means you're providing a selective pressure wherein only the recombinants grow and the non-recombinants are obviously being eliminated or being inhibited. And screening You are having a host of cells, a library of cells, and you are identifying only the recombinants. So in this video, we have been discussing about the selective measures, the selection methodology. So visual screening, insertion inactivation, which is involving an antibiotic resistant gene or a lac gene or the CI gene of the lambda vectors, fine. And then we saw with that of the complementation of mutation in the case of yeast vectors, with the marker genes like uracil 3, wherein you can have both the positive and negative selection. And then we conclude with that of a reporter gene assay, which is utilized in the eukaryotic cell lines. Fine. Thank you. And I'll get back to you with another lecture on the indirect screening methods, which involves the blotting techniques. Thank you.